Hello and welcome to my talk for this year's symposium. My name is Noemi Buisset and I'm from Medicover Genetics here in Berlin, where we recently conducted a study to detect and assess discrepancies between the short-term culture and the long-term culture of chorionic villi that we detected with OGM, and which I would now like to present to you. I will start by giving a quick overview of how the development of the problem for this project came about, our methodological approach, and then present our key findings. So the study originally arose from the idea to implement OGM from native villi as a first-tier analysis in CVS diagnostics. In CVS diagnostics, we analyze placental material exclusively, so more specifically chorionic villi, in order to then draw conclusions about chromosomal abnormalities in the fetus. And by implementing OGM, we were hoping to expand the diagnostic spectrum due to the higher resolution and the increased sensitivity compared to conventional chromosomal analysis, which is currently being performed. And also, we hope to be able to also detect smaller structural variants, as well as more complex structural rearrangements that are currently overlooked by conventional chromosomal analysis. And in addition to that, we hoped that by performing OGM exclusively from native villi, we might be able to decrease the turnaround time, as we would not have to wait for cell culture. Now, there were a few challenges that emerged from that idea, first being that to this date, there is no validated Bionano protocol for the DNA isolation from native villi. And second, as I already mentioned, we are analyzing the two cell layers of chorionic villi separately. So we analyze the cytotrophoblast in short-term culture and the mesenchymal core in long-term culture. And now we've observed discrepancies between the 2 and 3.2% of cases here in our lab. And these discrepancies can be indicators for CPM, which stands for confined placental mosaicism, and which refers to a type of mosaicism where the aberrant cell line is confined to the placenta only and does not affect the fetus. And because we are analyzing placental material in CVS diagnostics, these type of mosaics naturally pose a risk for incorrect diagnostics in regards to the fetus, especially if, as planned, OGM were to be performed exclusively from the directly prepared native villi, because the direct preparation does not separate between the two cell layers, and we estimate native chorionic villi to contain approximately 90% cytotrophoblast and 10% mesenchymal core, which obviously means that we would have a significant underrepresentation of the mesenchymal core. Therefore, our objective was not only to test the technical feasibility, so are we able to extract DNA from native villi and perform OGM, but also, and more importantly, we wanted to examine the diagnostic relevance of those discrepancies we would detect between direct preparation and long-term culture, especially in regards to the aberrations that would be confined to the mesenchymal core, and that would therefore only be detected in long-term culture and might possibly be overlooked if OGM were to be performed exclusively from native villi. And then finally, we wanted to put our results and our data into the scientific context of CPM research, because the majority of data and studies on the prevalence of CPM is being sourced from conventional chromosomal analysis and in rare cases microarray, which means that in turn they only refer to aneuploidies or large structural aberrations, but not to the type of aberrations that we would additionally be able to detect with OGM. So the study was primarily a proof of concept study using a very limited sample size. So we had 20 samples in total. And it was divided into a practical phase and an evaluation phase. In the practical phase, we performed the technical validation, which meant that we performed chromosomal analysis from short-term culture and long-term culture, and simultaneously extracted DNA from the native villi, which was our direct preparation, and the cells from long-term culture to then perform OGM. Afterwards, we compared the chromosomal analysis results to our OGM results in order to assess the concordance between the two methods. And finally, we analyzed 10 out of the 20 samples more in depth to reveal discrepancies between the direct preparation and the long-term culture in OGM. And for variant detection, we use two different algorithms, the de novo assembly and the rare variant pipeline, in order to detect both very small aberrations down to 500 base pair with the de novo assembly, but also low-level mosaicism down to an allele frequency of 5% with the rare variant pipeline. And we also hope that we might be able to detect that small amount of mesenchymal core that we estimated to be present in the direct preparation with the rare variant pipeline. Right, which brings us to our results. The technical validation was successful. We were able to extract DNA and perform OGM from 20 out of 21 native samples. And during the extractions, we noticed that there was a moderate positive correlation between the amount of villi we used for the extraction and the length of DNA molecules, which you can see in the diagram. So the more villi we used for the extractions, the longer were our DNA molecules, and therefore the higher our DNA quality. We were also able to establish optimal conditions for tissue input and storage, and we would recommend to extract DNA as soon as possible after arrival if you do not plan on freezing the tissue. Right, afterwards, as I said, we compared the two methods to assess the concordance, and there were two samples in which discrepancies between the short-term culture and the long-term culture were already detected in chromosomal analysis, and which were both not confirmed in OGM. First was in the short-term culture of sample 5, a Robertsonian translocation in 10 out of 15 counted metaphases, which resulted in a trisomy 13. And now Robertsonian translocations can generally not be detected by the method due to their breakpoint being in the centromeric region. However, the trisomy 13 should have technically been detected. Now, our most likely explanation is the distribution of the villi to short-term culture and direct preparation, meaning that as not all cells were affected by the aberration, what happened most likely was that the villi selected for the direct preparation did not contain the aberrant cell line, whereas the villi selected for the short-term culture did contain the aberration. And the second discrepancy was this time in long-term culture, also a mosaic, where we found a tetraploidy in 42 out of 50 metaphases and an inversion on chromosome 7 in the rest of the metaphases. Now, they were both not detected by OGM. Again, tetraploidies cannot in general be detected due to method-related limitations. However, inversions are usually very well detected by OGM. In this case, we had a similar issue where the breakpoints of the inversion were very close to the telomeric region and the centromeric region. And also due to because of the discrepancy between long-term culture and short-term culture, in this case, we performed amniocentesis, which confirmed a normal karyotype and therefore confirmed our OGM result, which ultimately led us to a concordance of 98% between the two methods. Now, in sample 3, chromosomal analysis detected an insertion of a fragment of chromosome 5 onto the long arm of chromosome 16, where with OGM, we were then able to demonstrate that that fragment was actually composed of multiple subfragments that were then reassembled in the wrong order and inserted as a single fragment back onto chromosome 16, which means that the breakpoint determination was significantly more precise using OGM than it was with using chromosomal analysis. Now, finally, we wanted to reveal discrepancies between the direct preparation and the long-term culture. So we analyzed 2,661 calls in total in both direct preparation and long-term culture. That was after the exclusion of artifacts. And from these, 44 calls were discrepant, which meant a concordance of 98% between direct preparation and long-term culture. And also, none of the 44 discrepancies would most likely have had any diagnostic consequences. And that includes the 20 discrepancies that were detected in long-term culture, because they either did not affect any genes or they only affected genes that had no disease association, the aberrations were intronic, or they only affected autosomal recessive genes, but the aberrations were not homozygote. And now I would like to present one of the 44 discrepancies. So a quick example, which was quite interesting to us, and which was a 300 kilobase per large deletion detected in the long-term culture with an allele frequency of 41% in the de novo assembly and a frequency of 27% in the rare variant pipeline. And that deletion was not detected in the direct preparation in the de novo assembly. However, we were able to detect the deletion with the rare variant pipeline in the direct preparation, which was very interesting to us because that meant that in this case, we were most likely able to detect that small amount of mesenchymal core that we in the beginning estimated to be present in the direct preparation with the rare variant pipeline. So that was very good. Right, and now to put our data into the scientific context of CPM. As I previously mentioned, most of the data on CPM is sourced from conventional chromosomal analysis and refers to aneuploidies and large structural variants. And the prevalence of CPM there is estimated to be around 1 to 2%. Now, we were able to detect discrepancies in all our samples, which equaled a prevalence of 100% of CPM if considering smaller structural variants. Also, most of our discrepancies were mosaic and smaller aberrations, so under 10 megabase pair. And these results were mostly concordant with a study published in Nature magazine in 2021, where Correns et al. Performed whole genome sequencing from 86 bulk placental samples and detected extensive mosaicism in placentas from single nucleotide variants. So they detected around 38 to 259 substitutions per placental sample, which obviously also equaled a prevalence of 100%. And they also showed that most of the substitutions were mosaic. Now, recently we found that we are not the only ones hoping to implement OGM into prenatal diagnostics, but that Gumi et al. In France published their findings in November following a prospective study where they performed OGM from 37 prenatal samples, including 11 chorionic villi cultures. They also compared their results to chromosomal analysis and to microarray. They found to have a 100% concordance and identified additional structural variants around 30 per sample that were smaller than 100 kilobase pair. They also assessed the clinical relevance of the SVs they detected in OGM in regards to the phenotype of the fetus. However, in that study, they only used cell culture from chorionic villi. So they published a second study where they, as we did, also performed OGM from native villi, from 6 native villi. In their case, they froze theirs prior to the extractions. They used the same protocol and the same kit that we did. And they also analyzed around the same metrics or parameters that we did. So their quality control metrics, the length of their DNA molecules, and they also tried to establish optimal conditions for their storage and preparation, which was very nice because we had something to compare our results to. The results were mostly concordant, especially in regards to the optimal conditions and the QC metrics. However, they did detect slightly less structural variants per sample than we did in the de novo assembly. However, that was most likely due to different filter settings. And they also said that none of their structural variants that they detected had any established pathogenic significance. Of course, our study focused more on the discrepancies between cytotrophoblast and mesenchymal core. So we differentiated between these two to better assess the risk of incorrect diagnosis due to CPM, which was important for us before establishing diagnostics from either one exclusively. And to summarize, we were very happy to find that the study showed the potential of OGM for using native villi in as a first-tier analysis in CVS diagnostics. because we had that high concordance not only between the methods, but also between the direct preparation and the long-term culture, so cytotrophoblast and mesenchymal core. Of course, you would have to mention that we had a very limited sample size, and we were also only analyzing extra embryonic material without comparing it to follow-up data. So the next step that we would like to take would be a randomized prospective study with a much larger sample size, where we would be able to compare our results to follow-up data, so either amniotic fluid or postnatal material. So thank you very much for listening to my talk, and goodbye.